So, hallo everybody. Good Abend. Ich heiße Flavia und ich bin Forscherin an MFPL Wien. Ich bin heute hier, um euch ein wenig über meine Laborarbeit zu erzählen. Da ich aus Brasilien komme und mein Deutsch nicht sehr gut ist, ich werde nun auf Englisch sprechen. So, hello again. Sorry for the terrible German. And I am here today, and I want to share with you all my love for science and virus. And maybe you are thinking, why are viruses so cool? So, first thing comes to my mind when I think about virus is how these little tiny microbes cause so much trouble inside cells, inside animals and plants, and so on. So this is what really fascinates me. This is what really intrigues me. And for that, I bring you some little friends that I have. And I want you to think that viruses are really, really small. And then we never see them unless we use microscopes. So if you have one idea, if you want to think about the size of a virus it's about 1,000 times smaller than the diameter of our hair. This is really small. So we really need to use microscopes to see them. So imagine you are everybody looking from microscope lenses, and then we have our virus. And we have so different viruses. For example, this one, I really like it because it's a microbe that infects another microbe. It's a virus that infects bacteria. This one here, coffee virus. It's a virus that infects coffee plants. Poliovirus, a virus that infects humans. And swine flu virus, a virus that infects pigs, as the name says, but also birds and humans. So as you could see, they have different shapes. And these are the differences we see from the outside of a virus when you look then through a really powerful microscope. But viruses can be also different from their inside. And for that, I brought for you today a really giant virus. So this is my virus. And I want you to imagine that if this virus has really this size, we humans, we should have at least 10 times higher than the highest mountains in the Alps. So it's really, yeah, so we have the size idea. And what I want you to think also, it's how a virus it's built, because they are really much more simple than we are. So for example, we have a body, we have skeleton, we have muscles, we have hair, skin, we have heart, while a virus is built mainly by proteins. So a virus is built mainly by proteins from this outside. So here we have these spike proteins, and this shell that's basically also made by proteins. And the inside, a virus is built from its genetic material, can be DNA or RNA, and also other proteins, for example, these ones. And what happens when a virus goes inside your cell? For that, I bring you today a cell, a really simple model of a cell. Then we see nucleus, the cytoplasm. Here for everybody, the nucleus and the cytoplasm, nucleus of the cell. And what happens when the virus infects the cell? So the virus really binds to your cell. So for this, I also want you to imagine that you are looking from microscopes. And this can be, for example, our skin cells. So the virus really binds to your cell. And what goes inside the cell, it's not the whole virus, as we see here. It's really kinds of breaks. And what really goes to your cell is the genetic material and some viral proteins. So they come here. And how viruses can reproduce? Because if you think they are so simple, so that's why they really need to go inside the cell. And when they are inside the cell, they are really producing new baby virus, and they are really mixed between the components of the cells. So we have virus here, we have the proteins of the cell, and we have the virus, the proteins of the cell, but also virus proteins here, here, and here. And that's why it's so important if you to study how the virus reproduce inside the cell and how we can differentiate them from the cell. And why is that? Because usually, of course, we want to kill virus if we got an infection. No one wants to have a viral infection. And then what you need to differentiate between the virus and the proteins of the cell or the whole cell. Because if you kill the cell, usually you cause harm to the human being you are trying to heal, so you don't want it. So you really want to fight only the virus when you have, for example, one antiviral drug. So it's really important to study it, and this is what I studied. So I really studied proteins of the cell and their properties. And let's think about properties as shapes and colors. So if you have the protein of the cell, the proteins of the virus, and proteins of the cell, and we know that they are different, so scientists can design antivirals that will bind to the protein of the cell and will inhibit the viral replication. The virus don't do new baby virus, and so the human being or the animal, it's healed from that infection. That's what we want. So I really hope you appreciate it and you understand, and thank you very much. Vielen Dank.